In this video, we are going to solve Cambridge O-Level 5090 paper 2 variant 2, most latest paper till now, May-June 2025, paper 2 for biology over there. Code is 5090. It is 80 marks question over paper over there. Before starting towards the solution of this paper, I will say kindly subscribe my channel, like the video and share the video. And it is a request, kindly subscribe the channel, okay? So that you can find the solution of the latest paper in a very explanatory style, and it will be much helpful for your preparation over there. Let's move to question number 1 there. Here he is saying a pea seed is planted in a pot containing soil and provided with the ideal conditions of germination. The pot is left in dark for 10 days. Okay. Shows day 1, day 2, day 3. Okay. And appears the germination over there. Day 1, day 2, day 3, X and Y. Describe the conditions required for the seed to germinate. Okay. Here I will say that conditions required for germination are including water, are including their oxygen, are including their suitable temperature. These are three conditions which must be satisfied for germination of seed over there. For water, I will say water will going to activate enzyme activation for and is to soften the seed coat. Water is required for enzyme activation. Enzyme activation and to soften the seed coat and to soften the seed coat over there. And after that, oxygen is required for aerobic respiration. Oxygen is required for aerobic respiration, aerobic respiration to provide energy for growth. Energy for growth over there. After that, he is saying suitable temperature for optimal enzyme activity. For optimal enzyme activity, suitable temperature is required. Usually warm or not extreme should be provided there, because these three are the basic conditions for the proper growth of seed over there. Now he is saying figure 1.1 shows that the seed changes day 1 and day 4. Describe these changes and explains what happened over there. So day 1 and day 4 is shown little bit over there. So I will say that the seed is originally lying as like day 1, and on day 4 what is this, that radical emerges first as a root tip seen on day 4 over there. This is the root tip over there, this is the radical, and seed coat splits as the embryo grows like that. Okay, let's write about this over there. What will I say? Radical emerges first, emerges first at day 4 as a root tip seen over there. Radical emerges first over there, and seed coat splits as the embryo grows. Splits as the embryo grows over there. And I can also add over here that food store in the seed in the form of cotyledons provide nutrients. Food store food store in the form of in the form of cotyledons provide nutrients for growth, provide nutrients for growth over there. And I can also add over here that it is done via respiration, via respiration over there. After that, I can say here that plumule or shoot may start to emerge. Plumule, emerge. Plumule is, as remember that radical is for a root and plumule is for shoot over there. Shoot, plumule emerge, plumule emerge to start plumule emerge, but it is not visible in early stages, but it is not visible in early stages over there. And we can also say water uptake causes causes swelling swelling and activate enzymes and activate enzyme enzyme that converts stored starch into glucose, stored starch into glucose for respiration. Ration over there. So these are the changes which are going to happen from day 1 to day 4, where a radical is emerging, where plumule is emerging, and water is showing a seed coat is also splitting up and swelling with the activates the enzyme and converts starch into glucose, which is required for respiration for the plant over there. In part C, figure 1.1 shows that after 10 days, the structures are labeled X and Y had developed from the parts of seed. So X and Y. So X and Y, X is basically a radical. X is the radical young root. This is Y. Y is a young root over there. And X is being shown as embryonic root over there. So I can say this is for stem and this is for a root over there for Y. So what he is asking over there, name the part of seed X has developed from. So X is upper part in the form of leaf over there. So it is plumule over there, which is responsible for making a shoot over there. The seed is germinating in darkness. Explain why X and Y to grow in opposite direction over there. So I will say that X, which is plumule, shows X, which is plumule, which shows the negative gravitropism like that, which shows negative gravitropism and Y as a radical as a root over there, radical as a root is going to show that it is going downward response, and which shows the positive gravitropism over there. And for this reason, I will say that in darkness, light is absent over there. As he said, that the seed is germinating in darkness over there. So I will also add here that in darkness, light is absent. Light is uh, sent there, and we can say gravity is the main conditional, and gravity is the main directional stimulus. The main directional stimulus. So if you go for like in the light, if it was happening, then I will say that maybe the shoots or plumules is going to show the positive phototropism and radical is showing negative tropism can involve over there. But here only one directional stimulus is gravity, so it should be taken like this. After that, okay, question number two, he is going to start with. He is saying that scientists have been thinking about how to feed the human population of the planet is in a healthy and sustainable way. They have produced produced model called how to feed the human population of the planet in a healthy and sustainable way. Okay, whole grains, starchy vegetables, dairy foods, animal sucrose proteins is also being taken. Plant sucrose protein, for example, beans and nuts and plant oils, and added sugars and vegetables and fruits over there. All right. So he is saying that the vegetables and fruits provide some protein, carbohydrates, lipids needed in a balanced diet. Name three other components in balanced diet. So if I talk in balanced diet, he said that proteins, carbohydrates, and lipids are needed. Three are already mentioned, so three more are required. Then I will say that is vitamins. And in case of vitamin, like he said that that can be obtained from vegetables and fruits, then I will say go with vitamin C that we get from citrus fruits over there. After that, minerals. And minerals, if I talk, let's suppose iron can be taken from spinach over there. After that, dietary fibers, like any roughage is going to provide dietary fibers in our food for balanced diet over there. After that, he is saying state two reasons why the plant's oils and other lipids are important in human diet. So for this one, I'm just writing the two main reasons that they provide fatty acids. They provide essential fatty acids, essential fatty acids needed for cell membrane, needed for cell membrane growth over there. Cell membrane. After that, I can say act. They can also act as energy stores, and absorb fat soluble vitamins and absorb fat soluble vitamins over there. So we can say that like fat soluble vitamins, if you remember about them, then they can have A, vitamin D, vitamin E, and vitamin K. All are these are fat soluble vitamins which are absorbed from by the fats over there. Okay, this was the main reason that plant oils and lipids are important in human diet over there. After that, he is saying table 2.1 shows that recommended daily average intake sources of protein for animals and plants in the planetary health. Okay, real meat, animal, poultry, animal eggs, animal fish, animal legumes, plant and nuts from plants. Recommending average intake in grams per day is given over there. So what he is asking what to do, use table 2.1 to calculate the percentage of the plant protein sucrose contribute to the recommended average daily protein intake for human space working. Okay, so total animal proteins is average intake. Okay, total protein sources given over there. So I will say they are having 14, 29, 13, 28. So first of all, I will add all of them. But I get 4, 9, 13, 13, 3, 16, 16, 8, 24. 4 is there to carry. 3, 4, 5, 6, 7, 8. So it is 84 gram per day. 84 gram per day over there is the total protein which is provided, and total protons is providing how much, 75 plus 50 is giving us 125 gram per day by the plant source over there. So if I add both of them like 84 plus 125, so 84 plus 125, 549 12 8 20 209 grams per day is required by the plants and animals over there. So I need to calculate percentage that plant protein sources contribute. So plant is 125, so I will say 125 divided by 209 multiplied by 100, then the percentage is approximately equal to 59.8%, which can be written as 60% over there is being taken with the help of plants over there. Now he is saying the global average daily human intake of red meat is approximately 300% of the recommended amount. Calculate how much red meat on average a human eats in 365 days. Give your answer in kilograms over there. So recommended is how much? Recommended is 14 over there, red meat. So recommended meat is 14 gram. And if I increase global average, then I can say it should go with 300% to increase. So 3 multiply 14 will equal to 42 gram per day. And he is asking that yearly how much he is taking in kg. Then yearly, and we know there are 365 days in an year, should be multiplied by 42, then I can get 15,330. 15,330. If I take in kilogram, then I will say it is 15.33 kg, which he is saying average yearly intake of red meat over there can be taken into account. Let's move to next question part over there. He is saying, is it is more energy efficient for humans to eat plants? Explain why. Because we can also say it is. I should go for environmental effect like that, like energy loss at each trophic level, loss at each trophic level when animal eat plants, when animal eat plants, 90% of energy, 90% of energy is lost as heat or waste, is lost as heat or waste over there. And I can also add that why this is energy efficient, because eating plants directly means human get energy from producers, reducing energy loss. So eating plants directly over there means human get energy from producers. Means humans getting energy from producers over there and reducing energy loss. So reducing energy loss over there. After that, he is saying if human reduce the amount of animals protein they eat, then less agricultural land is required. Suggest three environmental benefits of reducing this. So, okay, I'm just highlighting here that what they can get because they are getting animal protein they eat, then what will be the benefit according to environmental point of view? Then I will say there will be less deforestation, means human are not going to cut the trees over there. Reduce soil erosion, it reduce soil erosion, and less fertilizer runoff, and less fertilizer runoff into rivers over there. And I can also say that it will lower the greenhouse emissions over there. It will lower the greenhouse gas emissions over there. So it was related to mainly your biological molecules chapter, and also related to we can say agricultural point of view over there. Okay, question number three. He is going to okay. Now he is saying Staphylococcus aureus is a pathogen which infects humans. It can be described by antibodies and antibiotics. Okay, the cells of bacterium are spherical and about 1 micrometer in diameter. Staphylococcus S. aureus cell is shown as A, B, and capsule over there. Okay, name the parts A and B over there. So A and B. So I can say that A is just like a plasmid, and B is just like a mitochondria can be said over there. Okay. So it is plasmid, a small thin like points over there. And equal in size, I say these are mitochondria over there. After that, he is saying that part A can also be found in animals. Part A can also be found in animals and plant cells. State two other components of bacterial cells that can be found in plant cells over there. So it means we should check that what are these basically. So I will say A is a ribosome, not plasmid, is a ribosome. So he said what the others can be. So I will say that can be cell wall and cell membrane. Cell membrane is present in everywhere. Cell membrane and chromosomes. These two are present in animal cell and plant cell over there, and mitochondria is available and ribosome is available into all three bacteria, plants, and animal cells over there. Antibodies can destroy bacterial cells without destroying human cells over there. So explain how this is possible. I can say that antibodies bind to antigen, bind to antigen over there uh, on bacterial surface, on bacterial surface over there, which we can say this makes bacteria for destruction. This makes the bacteria for destruction by by phagocytes over there, and uh, it may also it may also neutralize toxins, toxins, toxins produced by bacteria, produced by bacteria over there. So this is the way how antibodies can destroy bacteria without destroying the human cell over there. Mhm. Okay, there are different types of strains SARS, and some can resist antibiotics better than others. A disc diffusion test. Okay, disc diffusion test is highlighted over here. Disc diffusion test is carried out to investigate the resistance of four strands of SARS. KLMN is shown below. Okay, in different antibiotics, the four strands of first grown that covers the surface of separate agar plates. Okay, the disc containing six different antibiotics are placed in agar plates. Six different antibiotics. Figure 3.2 shows the results of investigation after several days. Any clear zones indicate where S. aureus has died over there. So let's move what he is showing over there. He is showing strand K where agar plates are seen above. Okay. Disc containing antibiotics for strand L, strand M, strand N. So just why this is called disc diffusion test. So for this one, why it is called disc diffusion test, let's move towards it first part over there. Because antibiotic diffuses from diffuses from disc into agar, disc into over there, because it is showing the diffusion from disk to agar forming a concentration gradient, forming a concentration gradient over there. After that, describe why these results show about effectiveness of antibiotics 1 and 5 over there. So if we observe antibiotic 1 and 1 and 5. So I will say it shows for antibiotic 1 it shows large clear zone, large clear zones, large clear zones in all strands, and in all strands, which is showing large clear zones in all strands are shown, which we can say very effective against all over there. And for antibiotic 5, I will say small and no clear zones in strand, no clear zones in strands K, L, and M. I think so. These were three only over there. K, L, M. Okay. And we can say not effective or not resistance. Not effective, or we can say resistant over there. Okay, let's move to the next part over there where he is saying over time the percentage of each strain is the Auris Aries population will change. So just which strain KLMN in figure 3.2 is likely to become the largest percentage of the population over there. So I will say strain N, I think so. Strain N. So he is saying explain why the strand named in C1 part is likely to become the largest percentage of the population over time. So I will say it will be going towards natural selection. Natural selection over there, in which I can say natural selection like antibiotics kill the susceptible strand KLM as natural selection as I can say antibiotics is going to kills susceptible strands like K, L, and M over there, and the resistant strain N survives and reproduce without competition, and strain N survives and reproduce without any competition over there, without any competition, and passes resistance genes to offsprings, and passes resistance to offsprings. So their population is going to increase in that section over there. Okay, let's move to question number 4 now. Figure 4.1 shows the human bronchi and some of the specialized cells from the inner layer that surrounds the lumen. Okay, lumen is this, and inner layer, and X and Y are shown over there. So he is saying what? Name the cell labeled as X. So I will say X is a goblet cells over there. What I will say X is goblet cell. What is their function over there? Then I will say it produces mucus. It produces mucus over there, mucus, and to trap particles and microbes. to trap particles and microorganisms or microbes. Over there, after that he said structure Y. Yeah, structure phi, I will say ciliated epithelial cell is ciliated epithelial cell over there. So its function is like we can say beats it beats mucus upward to trap and remove to trap and remove remove um, dust or pathogens from airways. From airways over there, and this is the function for ciliated epithelial cells over there. Now he is saying that the muscle layer in the walls of the bronchi, the muscle layer in the walls of the bronchi and bronchioles sometimes contracts, causing the lumen to narrow. Okay, this is called constriction. Okay, bronchoconstriction. And on one substance that triggers the contraction is nicotine over there. Okay, what he is asking, suggest the effect of bronchoconstrictions on a person over there. All right, so he is asking for 3 marks. So I will say effects first of all. If I go with he is asking for 3 marks. Okay, I will say it will narrow. It will narrows the airways. It will narrows the airways and it will reduce the ventilation. Reduce the ventilations over there, and I can also say it lowers the gas exchange efficiency. It lowers the gas exchange efficiency. After that, he is saying describe one effect of nicotine on circulatory system. So I will say it increases the heart rate and blood pressure. The heart rate and blood pressure over there. So this is the way we can say it stimulates the adrenaline release over there to make it more harmful or like that. Okay, let's move to next question here. Here he is saying that rhizomucor uh, bacillus is a fungus found in the piles of dead plants. Okay, vegetation. It has an optimum growth rate between 60 to 70 degrees centigrade. Identify and name the kingdom of the genus of this organization over there. So according to this, I will say kingdom is fungi and kingdom is fungi and genus can be taken as rhizo because this is a genus. According to scientific name, we use the first letter from the genus over there. So I will say it is rhizomucor. What I am telling you, when scientific name is being taken, the first letter is taken from the genus and kingdom is being taken according to fungus or fungi over there. After that, he is saying give two of the main features of the organisms classified in the kingdom named as fungi over there. So, like I can basically say their cell wall is made up of chitin over there. This is the main characteristic of fungus. If we take for plant cell wall, that is made up of cellulose. If we take for bacteria, that is made up of peptidoglycan over there. All right. And for their reproduction, I will say they reproduce by spores. Over there, these are the two main features of the organisms that are classified in the fungi kingdom over there. After that, he is saying explain how piles of dead plant vegetation can have the temperature much higher than the surrounding environment over there. Okay, let's go for this. I will say that there is a microbial respiration, microbial respiration is going to happen, which microbial respiration decomposes organic matter of dead plants and animals. Organic matter, which we can say it is exothermic reaction, and it is exothermic reaction which releases heat over there. Reactions which release heat over there, and insulation by pile reduces heat loss, and insulation by piles reduces heat loss. Okay, this is the complete section which he was expecting us, that how piles of dead plant vegetation can have the temperature much higher than the surrounding environment in the form of 60 degree to 70 degree centigrade over there. Okay, here he has shown some. The fungus produced by enzyme is called pectinase. The scientist extracted the pectinase from the fungus and investigated the pH on the temperature at the activity of 20 degrees centigrade, and activity rate is being shown maximum, as the value can be taken as, so it is 4.5 over there. So enzyme pectinase, where it is showing its maximum activity, then I can say activity increases from pH 3 to pH 3 point till 5, no, till 4.5 it is increasing. So optimum pH is around 4.5 to 5 over there, as showing the range, and pH above 6 is showing the actively decreasing, or above 5 is going to show decreasing rate over there. So pH affects the enzyme activity, active site shape, and ionic bonds are going to be removed over there. So the point which I want to explain you here, if we look towards this graph, what he is asking for this, explain the results of pH range from 2 to 7. So 2 to 7, if I want to explain, then I will say 2 to 3, 0. Then it is getting optimum pH. Then it is increasing till 4.5 over there. And after 4.5, we can say they are going to decrease and getting them to 7 over there. And we can easily take. So we can write about this when he is saying 2 to 7. Then I will say that from 2 to 3, 2 to 3, it stays 0, stays 0, and from 3 to 5, 3 to 5, it increases, it increases its enzyme activity as rate of reaction, and optimum pH is around, we can say, uh, 4 to 5 over there. So up 3 to 5 it increases, and optimum pH is 4 to 5, 4 to 5 there, and we can say above 5 activity decreases as enzyme denatures, and above 5 activity decreases as enzyme denatures over there. After that, he is saying the experiment was repeated at the temperature of 55 degrees centigrade. On the figure 5.1, draw a graph which suggests the results expected at 55 degrees centigrade over there. 55 degrees centigrade. So he has given me the same graph over there only. So we will have to draw the same activity, but I will say that I will draw like in the start it will be going to increase, and if it is 55 degrees centigrade showing optimum, and then again it should be showing the decreasing over here. So draw the curve shifted to the right with higher peak and closer to the fungus optimum temperature, and actively higher at 55 degrees centigrade than 22 degrees centigrade or 20 degrees centigrade, which he has drawn already. So and we can say that actively higher at 55 degrees centigrade than 20 degrees centigrade over there. After that, he is saying name an industrial process that uses pectinase over there. Then I will say industrial use. For this one, I should write about this, that it is clarifying fruit juices, like breaks down the pectin in cell walls over there. So I will say it is used as clarifying fruit juices, fruit juices to break down the cell walls, breaks down pectin in cell walls, break down pectin in cell walls over there. So it was the use of this process over there. After that, he is saying that he has shown the nucleus of the cell contains DNA. Nucleus of cell contains the DNA molecule and that controls cell function. Figure 6.1 is the diagram showing one of these DNA molecules over there. Okay, and cytosine, guanine is being shown over there, and Q is shown over there. Okay, he is showing, he is saying state the name of the structure labeled as P over there. So P is just like a chromosome structure over there, I can say. It is just like a chromosome over there. And state the name of the unit of DNA labeled as Q. So that is a nucleotide. Nucleotide over there. And state the letter with the base pair of G will be, as we know, adenine is making double bond with thiamine and cytosine is making triple bond with guanine. So it is cytosine. over there. And in the part B, explain how this DNA molecule controls cell functions. Include an example in your answer over there. So I will say that DNA, DNA, deoxyribonucleic acid contains genes that code for protein, that code for protein, and via transcription and translation, via transcription and translation over there. And we can say, for example, I am saying gene for insulin, gene for insulin production over there. But insulin is just like a protein. Then gene for insulin will communicate towards messenger RNA, and insulin protein, and then insulin protein insulin protein regulates blood glucose, regulates blood glucose, and controls the enzyme production, controls the enzyme production, controls the enzyme production over there, which controls the metabolism, and mutations can change the protein function, which will affect the cell actively over there. All right, so you can add this, and you can also add over there that it controls the enzyme production by controlling metabolism, controlling metabolism, and mutation can change, mutation can change mutation can change protein function, protein function protein function, which can affect cell actively, which can affect which can affect cell actively. So it was your whole detail related to the DNA molecule control over cell functions over there. Okay, so this is the question number 7 there now. He is saying photosynthesis and transpiration are two processes that take place in plant leaf. Explain how the structure of leaf is adapted for photosynthesis. So I will say the adaptations by the leaves can be written as broad and thin shape, large surface area for light absorption. So it can say broad. Broad means wide. Broad, thin shape. Thin shape is the first adaptation to the leaf. For large surface area for thin shape, and large surface area for light absorption, light absorption over there. And we can say that chloroplast in palisade layer, and number 2 I will add here, chloroplast chloroplast in the palisade mesophyll layer. In there are two type of mesophyll cells, palisade mesophyll cells and spongy mesophyll cells, but right now I am saying chloroplast in palisade mesophyll layer, mesophyll layer in mesophyll layer, near to the top, near top for maximum light, near top for maximum light over there. And we can say that stomata allow carries entry, and stomata allows or carry entry, allow carbon dioxide entry, carbon dioxide entry over there. And we can also say air spaces in spongy and air spaces in spongy mesophils, in spongy mesophil cells, spongy mesophil cells are available for air spaces in spongy mesophil layer or cells over there, spaces for gas diffusion is available over there. And I can also say that network of veins and a of veins network of veins transport water minerals, transport water and minerals and to remove sugars, and transport water and minerals and remove sugars, remove sugars over there. So I can also add over here that there is a waxy cuticle. There is a waxy cuticle, which reduces water loss, which reduces water loss, but transparent to light. So these were little bit adaptations which leaf gets for transpiration and photosynthesis to help out there. After that, he is saying transpiration is both necessary for plants and problem for plants. Discuss this statement over there. So I will say as necessity, first of all, I am saying as necessity that it creates, like transpiration creates a transpiration pull, transpiration pull for water and minerals, for water or minerals, water and minerals, uptake from roots, uptake from roots, and it cools, it provide cooling, it cools plant via we can say via evaporating cooling, like that. So it cools plants via evaporation. These were the necessities over there. And if I talk as disadvantages for this, then I can say it can lead to as disadvantage over there, or as pros over there. These were consequences and these were pros over there. So for these disadvantage, I can say it can lead to, uh, water stress or wilting, water stress or wilting, and for water for if water loss exceeds uptake, if water loss loss exceeds uptake, and it may cause stomatal closure, stomatal closure. It may cause stomatal closure by reducing photosynthesis. By reducing photosynthesis over there. Okay. Okay, it was the end of your paper completely over there. I tried to explain as much as good for you. Its paper one is also attached in this, we can say, playlist. So kindly subscribe the channel so that you can find the more detailed solution of all the papers over there, and share the video, like the videos, and if you need any paper solution over there, kindly comment below. I will try to upload soon as I get the time for your help in your final examinations over there. Thank you so much for today.